This video, we will be discussing the very exciting molecule known as RNA, or ribonucleic acid. Ribonucleic acid is necessary in your cells, extremely necessary, so that your DNA doesn't have to leave the nucleus, because when DNA leaves, it could be mutated. So, RNA is to move information from your DNA into the cytosol so that proteins could be made. Basically, the messenger of DNA to the rest of the cell. So RNA is structurally very similar to DNA with specific differences. First of all, the sugar molecule is known as ribose, not deoxyribose. There is a nitrogen base known as uracil instead of thymine, and they're mostly single-stranded, not double-stranded. So you generally get one single strand, whereas DNA, if you remember, has those two strands. And then lastly, there's more than one type of RNA. There's actually 3 different types of RNA. Those 3 different types of RNA are messenger RNA, also known as mRNA, which is, as it's actually named, a messenger of DNA to the rest of the cell. So it carries the genetic information from DNA to the cytosol. tRNA, transfer RNA, is RNA that actually carries around an amino acid, which, if you remember, amino acids are the building block of proteins. So the transfer RNA is required to make sure that the amino acids, the correct amino acids, come and form the correct proteins. And then the last one, ribosomal RNA, is the RNA that is found in the ribosome, and it basically reads the messenger RNA in order to make a protein. So each one of these different types of RNA has a specific function in the cell. Now, how you actually make this RNA is a whole different process. The process is known as transcription. Transcription is copying the genetic code from DNA to RNA, and it is composed of 3 major steps. And it has one major enzyme known as RNA polymerase. So the first step is initiation. And initiation is when RNA polymerase comes and binds to the DNA molecule at what's known as the promoter. So it's RNA polymerase coming in and splitting your DNA molecule into 2 strands. The promoter is a start of a gene, and it's known as kind of the starting line for transcription to take place. And it's a little signal and a little location where the RNA polymerase knows to attach. So it starts there and moves along your DNA, making your RNA. So it starts to split the DNA so that the RNA can come in and be formed. And only one strand of DNA is actually used. It's known as the template strand. The other strand is known as the non-template strand. But the template strand is very important, and the fact that there's only one template strand on a DNA molecule is very important, because if not, then you would be getting a different RNA molecule every single time that you transcribe the gene, and therefore a different protein, which would cause all sorts of problems in your body. The second stage in transcription is known as elongation. And elongation is basically the RNA polymerase moving along the DNA and adding complementary nucleotides. And A pairs with U and not T. And the RNA grows in the 5' to 3' end. Same idea as DNA replication. If you remember 5 to 3, 5 to 3. RNA has to grow the same way because polymerase functions the same way, whether it's RNA or DNA, and will only add to the 3' end. So it continues growing and elongating, getting longer, until it reaches the termination signal. The termination signal is like the finish line, and it marks the end of a gene. So you have the promoter on one side of the gene and the termination signal on the other, and that makes sure that only that gene is transcribed, because this only occurs when your cell needs that particular protein that that gene codes for. So that's where the whole control of this comes into play. Your DNA controls what's being transcribed, therefore what proteins are being made. So at the end, the polymerase detaches, and you end up with your DNA, which is back to normal, and your RNA molecule. So what you actually end up from all of this are all 3 types of RNA, and it depends on the polymerase that is used as to which type of RNA you're going to get, whether you're going to get T, M, or rRNA. Now, majority of the time, you're getting the mRNA, because when we get into actually translation of this, which means taking that RNA and making a protein, you'll see that tRNA and rRNA can get reused, whereas mRNA cannot. So mRNA needs to be made every single time that protein is needed, every time that gene wants to be expressed. So that's the sum of actually making RNA. The next step in this, as I said, is translation, and that's going to take that RNA nucleotide sequence and make it into an amino acid sequence. So if you think transcription is just copying things down, so transcription is DNA being copied into RNA, and then translation is actually taking that copy and changing it into a different language or the amino acid sequence. So that's going to be the next step, and all of this is coming around gene expression. It's all about how to express genes and when they're expressed, how they're expressed, why they're expressed, and generally they're expressed when necessary. So this all goes back to how you actually control your genes, which is from your DNA.